Our next speaker is Dr. Subhabrata CHAKRABARTI. He will be speaking on role of genetics in prognosticating of primary congenital glaucoma. Dr. Subhabrata is in the genetics department at the L.B. Prasad Eye Institute for more than 25 years. We have the world's largest series included in a large cohort of primary congenital glaucoma genetics. He will be talking on that. Thank you.
Thank you, Dr. Mandal, for that very generous introduction and also for the opportunity to speak here. So the topic that I was given is role of genetics in prognostication of primary congenital glaucoma. As you know that it is not only complex disease, but it also has a very multifactorial etiology that when I go through. Debashish and Murli have discussed in great detail about the phenotypes and how there could be overlaps, how there could be challenges in diagnosis. Now, what about prognosis? So, prior to that, yeah, there's the customary that I don't have any financial disclosures. So, for a patient, what we are looking into, we need to kind of link the genotype to the phenotype. So, an undiagnosed patient comes to your clinic, and we need a very structured phenotype. That means the phenotype of the patient has to be in a way that it falls under a specific diagnostic regime. Now, with the advent of genomic technologies, one could go in for identifying specific candidate genes, given that there are genotype-phenotype correlations for certain genes. In other cases, it becomes goes on a research mode in terms of clinical sequencing, in terms of sequencing of the exome or the whole genome, and the corresponding family members to generate the data on inheritance. The data is then shared, the genotypes are linked to the phenotype along with the data. In the background of course, the ethical considerations are there. But since we are doing, we are kind of undertaking whole exome or whole genome sequencing, additionally that needs to be validated in appropriate model systems. For instance, it could be an in vitro model where one actually introduces those variations seen in the genome in particular cells or primary cells or cell lines, or it could be in vivo models using a particular animal models. We don't have any specific animal models for glaucoma. What we do have is models for quantitative traits, like we have models for intraocular pressure, we have models for enlargement of the diameter and enlargement of the globe particularly, and so we try to kind of correlate those, and based on that one again comes to a particular diagnosis to see whether that kind of links to the genotype, and thereby whether that can change management. If there is a perfect genotype-phenotype correlation, whether that can change the practice pattern, that can change the management or the future planning, as Murli highlighted too. So, in terms of from a clinical perspective, it is more of genetic testing, and as we know that there are two modalities of testing. One is the diagnostic testing, where one tries to establish a particular link between a genotype and a phenotype. The other is predictive testing. Now, in diseases like congenital glaucoma, where one is kind of establishing a diagnosis, we really don't go for predictive testing, because in these cases, the disease has already set in, it is congenital. Predictive testings are mainly done either for a pre-symptomatic condition where you have a family where the individual is symptomatic and hasn't developed the symptoms yet, or they are predisposed because they have a family history of the disease. In either of these cases, these are mainly the age-related conditions where this thing is kind of seen, and in terms of congenital glaucoma, it will be more of looking into the diagnostic modality. So, currently there are multiple genes that have been identified in congenital glaucoma. As both of the earlier speakers said, that CYP1B1 or the cytochrome P450, which is an enzyme which has been linked to the disease, not necessarily it is causal for the disease, and that has a wide range of prevalence across different populations. And if you look into populations both from India and other places, there are populations which have higher prevalence of these variations in this gene, but not necessarily these are causal. and the other genes that were simultaneously mapped, like a solute transporter LTBP2 or an angiopoietin receptor TEC, you would see that in the Indian scenario, these have much lower penetrance, much lower prevalence, as well as much lower effects on causality. But what happens with the cytochrome P450, the reason that it was mapped is that there are certain signatures in cytochrome P450, and no matter from which population you come from, you harbor that signature. These signatures are basically, if you align those variants on a chromosome, on a single or a haploid chromosome, we get something called as a haplotype. So mutations are structured on these haplotypes, which means that if you have a mutation, let us say it's the glycine product, glycine to a glutamic acid, and it's in patient in Ecuador, whether you are from Morocco, Saudi Arabia, or India, you will harbor the same haplotype. Meaning thereby this, since evolution, since the migration of populations, these haplotypes have been constant. And so these are good for kind of understanding that they have a disease which is linked to this, but this does not necessarily mean that they would, they would conform to primary congenital glaucoma, because even in other conditions, even in individuals who are asymptomatic, you might find such kind of haplotypes. Now, what about genotype-phenotype correlations? So, for patients who are harboring those kind of changes, and if we look into the quantitative traits like intraocular pressure, corneal diameter, or cup-to-disc ratio over a period of time, and given that they are harboring 2 copies of the mutant allele or 1 copy of the mutant allele, or do not have any mutant copy, there is practically no relations with CYP1B1, and this is a data that we combined our data with the Brazilian data. It was over 950 patients from both the countries. And what we show is there is a minor involvement of poor prognosis only with homozygous CYP1B1 variations. But where we did find some kind of an evidence is a variant in the promoter region that regulates the functions of CYP1B1, and this region harbors a variant which is strongly associated with primary congenital glaucoma cases. Given that there is this variant, it will reduce the activity of CYP1B1, and CYP1B1 is required not only for development, but it actually converts retinoic acid, it regulates the retinoic acid signaling pathway, converts retinol, which is an alcohol, to an aldehyde, which is retinal, and in the absence of CYP1B1, the developmental process is impaired. So that's one of the clues that we have. And given that they have this variation, the reduction of CYP1B1 activity occurs to the tune of 90%, and there are other transcription factors which are involved in development, like the GATA1, PBX, etc., and their bindings are also impaired. But from a clinical perspective, now the children who are harboring this, and if you are following them up for an year, the child who harbors this variant, they also have corresponding variants in the coding region. That is why they have a haplotype. And if you see that in terms of their prognosis, in terms of their IOP control, they have poor IOP control compared to those who do not harbor those variations. So that's one of the only indications in CYP1B1 that if one looks into mutations in the coding region, that has to be in consonance with the promoter variant. So other than that, we now have ClinGen, which is another organization as per the NIH's directive to kind of integrate all this data, genomic and health data, for improving genomic medicine. So the variants that are observed are obviously, they are characterized on the basis of multiple other parameters, on the basis of screening of controls. And in the glaucoma variant curation panel, those of us who kind of are involved in this, what we try to do is to see that if there are variants that are uploaded, how many of them would actually have a clinical implication to show the impact of those variant on the phenotype. And that is the database that we kind of manage. And what we have shown is that even for changes which leads to the truncation of the protein, there are certain parameters which have now been improvised, and it's not only the presence of the variations, but it is also the function of those variants. Do they actually regulate the function? Is there a reduction of function? And so on and so forth. So if one goes in to identify those under a process of genetic testing, what we find that for the candidate genes, there will be around 20 to 30% of the variants. So if you concentrate on the purple part, a larger part of the section will be unknown. If we target other genes, for instance, in the anterior segment and all other developmental genes along with the glaucoma-associated genes, that number of unknowns might reduce by 10% when we do whole exomes. It might reduce further, but doesn't necessarily take away the entire thing. That is because there are a lot of co-occurring alleles, which means that variant of one gene co-occurring with another gene, and they are functionally regulated. And this is not only true for glaucoma, even in retinal diseases, for instance. If we scan the entire genome, you see that around 30%, here it is around 32%, of the variants are always unknown. So when we undertake these functional assessments, what we see is if there are multiple variant of one gene coming from father, another from the mother, they are asymptomatic, but child inheriting both in a digenic fashion or in a multi-allelic fashion, they manifest the phenotype. We go on to assess their function and see how that impacts the protein function. So just if you could see that CYP1B1 interacts with TEC, and by themselves they interact, but if there is a mutation, the mutation perturbs that interaction. The interaction comes down, and that is where the functions are perturbed. And in case of in case of the variant itself, like CYP1B1, like the heterozygous CYP1B1 variant, for instance, they do not interact and they do not impair any functional assessment. Likewise, there are other genes like serine protease, etc., where if you knock out those genes, they also interact with CYP1B1. If you knock out those genes in the animal models, like in the mice models, for instance, if you see the trabecular meshwork, you follow them over a period of time, the trabecular meshwork becomes hypoplastic as opposed to the normal one. And likewise in GLIS, which is another cripple transcription factor, if you follow them, if you knock out those genes because they interact with CYP1B1, the trabecular meshwork becomes hypoplastic. But how are we going now that we have this new data? How do we then take on genotype-phenotype correlations? So we used the photopic negative responses of these kids and compared to their fathers, compared to their parents. And what we find is that given that they have multiple variants, the photopic negative response in the child over a period of time is diminished. And if we quantitatively look into those, for instance, those who harbored those multiple variants, you see a stronger correlations of the clinical parameters in terms of their corneal diameter to the photopic negative response, in terms of their intraocular pressure control to various parameters of photopic negative response, as opposed to if you see on this panel that there are no changes, there are very minimal correlations for those who do not harbor those variants. Again, meaning bringing the fact that we need to see it on a multi-allelic scale and then try to kind of correlate the genotype with the phenotype. So there are different models, different pathways that we have proposed. One is the developmental pathway, the cellular death pathway contributing to PCG, and these are again individually contributed by individual gene variants from different genes and different pathways that interact together. One of the last things that I want to stress upon is, actually I have a talk again at 11:30, and some of the slides will be there as well, where we use multiomics to kind of characterize the disease. And what we have shown is that the presence of controls, a very deeply phenotype controls, is essential for understanding the disease phenotype, because these interactions also occurs in the normal individuals. There might be modifiers, but that needs to be eliminated. For instance, if we take the 1000 genome data or the GenoMat data, which is normally done by many service providers, what happens is, like, we have longitudinal cohort data where for across all the glaucoma and the anterior segment disease genes, we found that there are close to 400 rare variants which are causal. of which 143 are present in the general population. And these are deeply phenotyped since they are followed up over 3 times in 15 to 20 years. Now, if you see the distribution of those variants in the 1000 genome and other global databases, there are only 28 in Punjabis of Lahore, 28 again in Gujaratis of Houston. In Telugus, they have only around 13. Among the Sri Lankan Tamils, around 32. Among the Bengalis, around 23. So, if you take the representative of the population, there are so many less variants in the general population in the global databases. So, that will render your data to be false positive. Why? Because we see that in the general population, we have 143 if you take a local cohort. But if you use the global databases, if these variants are outside this 23 or 24 variants, every other variant will be considered as normal. So, that is where one needs to be kind of cognizant and vigilant about these facts. So, the key takeaways are that it is a complex disease. It is becoming complex. It used to be a Mendelian disease. But now we know that it is attributable to multiple gene variants with varying magnitudes of effect. And with a multifactorial etiology, the dissections would involve considerations of the multi-omic components. I will be discussing that in the 11:30 session, so I am not much of it. Genetic testing components are still varied and universal signatures are still being studied. So, just given that they have a CYP1B1 mutation may not always render them to be having PCG, there could be other conditions as well. Large effect alleles sometimes exhibit associations like we kind of find out sometimes, but they may be inconclusive for a genetic diagnosis. And as of now, even clinical diagnosis holds the key towards prognostication as the biological components for all these quantitative traits are unearthed. So, I'd like to thank all the patients and volunteers. Most of all the patients almost are from him, and the team at my end and the collaborators and funding agencies. Thank you.
Thank you. Thank you, Subho, for extensive presentation on